Good morning, class. I just want to say thank you so much for coming to Saturday school. So this is a short video to support your learning and for your upcoming Unit 4 exam on Friday. So we're going to start off with our first lesson, which we had, which was the membrane structure, right? So our cell membrane is the one that controls what goes in and out of the cell. It is made up of what is called a phospholipid, right? So phospholipids look like this. It has a head and a tail. The heads are called hydrophilic. Philic means loving, and hydro is water. So the head of the phospholipid is water loving. Here is the outside of the cell, and here is the inside of the cell. They both have water, so that's why the head is facing outwards. The second structure is the tail, so hydrophobic tail. So again, hydro is water, and phobic means fearing. It doesn't like water. It all wants to be away from water. This is why your tails face each other. They don't want to face water, so they avoid it as much as they can. That's why it is called a phospholipid, because it is made up of phospholipids, and bilayer. It means two. So each one, here's your top layer, that's layer one, and then your second layer is down here. So that's why it's called a phospholipid bilayer. Normally, very small particles can go through your phospholipid bilayer very easily, but then you have larger molecules like glucose that need to use a channel protein. So again, channel proteins are these big structures that help larger proteins pass through the membrane. For ATP, you have what it's called carrier proteins. So again, they also help in the process of moving particles across the membrane. So again, the phospholipid bilayer controls what enters and exits the cell. So remember, members only, in and out. It is found in all cells, prokaryotes and eukaryotes. So every single living thing has cells that has this structure called a membrane, and it is composed of phospholipids. We already saw that it's called the phospholipid bilayer, and proteins. Remember, these proteins let large particles go through, or they facilitate in active transport. This next one, class, is concentration. So these little particles that you see here are your solute particles. Solute is the one, or the what is being mixed in. So if you're making Kool-Aid would be these little particles, and the rest is like water, your solvent. Remember, high concentration means that it is more crowded. So if you see here, there is a lot more purple dots. And then low concentration means there's less. Naturally, you go from crowded to less crowded. Naturally, this is going with your concentration gradient, going from high to low. On the other hand, when you go against the concentration gradient, you're going from low to high. You're going into the crowd, so you need energy. This is specific for active transport. If you go from high to low, which is what you normally see, that's passive. Again, equal, that word comes back. We always want to be in equilibrium. So eventually that's where you want to get, where both sides are in equal concentration or equilibrium, or also homeostasis. So again, concentration is the amount of solute particles, and you try to go from high to low if you're passive, and if you're active, you go from low to high. You go into the crowd, so you require energy. Here it is a quick comparison. So that A in active should tell you this first one, that it uses ATP. Osmosis, remember, osmosis is specifically the movement of water from high to low concentration. So it would be considered passive transport. Diffusion, any type of diffusion. Remember, diffusion again goes from high to low concentration. So that is considered passive transport. Molecules move across the cell membrane. That happens in both. So this one will be in both. Molecules move across the cell membrane in active transport and passive transport. They're both transport. Facilitated diffusion. Remember, facilitated diffusion is a type of diffusion where your particles, like glucose, are a little bit too big. So they use that protein to go through the cell membrane. Moving your molecules from high to low, that is passive transport. Remember, going from high to low, from crowded to less crowded or uncrowded, that is going with the concentration gradient. Endocytosis and exocytosis. We just learned that. So endo means you're bringing something inside, and exocytosis means it is being exited. That tosis should be a clue that you are in ATP. So think of the T or whatever connection you can make. But endo and exocytosis are active transport. Both help maintain homeostasis. Both of these. That is the whole goal of our bodies, to be in equilibrium and homeostasis. So both of them, both active and passive transport, do that. No ATP. So no energy would be passive. No más se pasa. No energy is required. And lastly, particles from low to high. This is your active transport. Because you're going into the crowd. Imagine having to push yourself to get to like the front of a stage at a concert. A lot of energy wasted. So again, active and passive, comparing them will be on there. Make sure to study the notes, and we'll continue. This next one is just showing you little images. So remember, diffusion. Simple diffusion is when particles, this is high concentration because there's more solute, there's more purple dots to low. And it happens fairly easy. These particles are small. They just pass through the bilipid layer. Facilitated diffusion, this blue thing is helping your particles go through. So these triangles are probably like sodium or some large triangle. You're still going from high to low, but you're using that protein channel to help pass through. Remember, osmosis, this is H2O, specifically the movement of water. So you go from high to low. Again, I know the particles look a little bit different, but basically there's more solute. And the particles you see there are water. So again, you're going, the water is moving. All of these three, diffusion, facilitated diffusion, and osmosis, all of these are called passive transport. Remember, passive transport does not require any energy. And lastly, you have a generic active transport. You're going from low to high concentration. The big giveaway, this is called like a carrier protein or a protein pump, is that ATP. Whenever you see ATP, think of active energy. You're using energy. Your next one, class, this was the last topic we learned on cell transport. So endocytosis and exocytosis. Endocytosis means within or to bring inside. And exocytosis, you should remember that it looks something like exit. It is going to expel this. So endocytosis and exocytosis, both of these are active. Active transport, so you require energy. Again, in endocytosis, think of an endoscopy. You're going into your body. So that's what's happening here. These little particles get into the cell membrane and they go into the cell. So intracellular means inside, extracellular means outside. On the other hand, in exocytosis, sometimes something is inside the cell and it kicks it out. Normally it's hormones or waste that they don't need. So again, endo means to go inside. Think of an endoscopy. They put a little video camera down your digestive system, right? So that's endocytosis. It goes inside. We are inside the cell. On the other hand, exocytosis is kicking something out, whether it's hormones or waste. Again, these are active transport. And then lastly, you will definitely be tested on this. 3 to 4 questions. Hypotonic, hypertonic, and isotonic. This is under osmosis. So these are passive transport. It does not require energy. So again, in this image, you have water going into the cell. Whenever water goes into the cell, this is called hypotonic or hypotonic. I actually misspelled that. This is called hypotonic. In a hypotonic solution, the cell will swell or burst, and there is more solute inside the cell. Remember, water follows the party, so there's probably more dots here inside the cell, and that's why the water is going in. On the other hand, this one you have arrows going out and in. This is called isotonic. Isotonic means it's equal. So how many ever particles are on the outside, it's on the inside. So that with the being equal means there is no change in the cell, which is good. It's balanced. You don't have to worry about anything happening. Our last one, remember our arrows pointing out. You think of that hyper little kid. So this is a hypertonic solution. In a hypertonic solution, there is way more solute on the outside or on the actual solution than instead of inside the cell. So that's why water goes out. So again, the cell, because it's losing water, the cell will shrink. And water moves out. So again, when you see the word hypertonic, think of a hyper little kid that needs to go play outside. These are called tonicity. You have to know how the water moves and how it looks. That'll help you out a lot for Unit 4. So again, active and passive transport. For passive transport, we learned that it requires no energy. So it just passes through. There's no energy. Passive transport, you're moving from high concentration to low concentration, which is called with the concentration gradient. And we learned several types. The first one was simple diffusion, where small particles just pass through super easily. The second type was facilitated diffusion. So remember, facilitated diffusion is when you need a protein to help pass those larger particles. And the last one is osmosis. So remember, under osmosis, we have hypertonic. That hypertonic means water moves out, so the cell shrinks. We have hypotonic. Think of a hippo that swells, so water is going to go into the cell. And the last one is isotonic. It's equal and just right. Back to the types, then we have active transport. Remember, active transport, you have the sodium-potassium pump. Your sodium-potassium pump moves the particles through a pump with ATP. You learned endocytosis. Remember, endo means within, so endocytosis. With endocytosis, you learned phagocytosis. So phagocytosis is like the cell eating. So think of that pH sound. So it's like the cell is eating food. The particles that move into the cell are solid. Then you had pinocytosis. Pinocytosis is like the cell is drinking. So what moves in is liquid, like extracellular fluid. And the last type, remember, was exocytosis. Exo for exit. So this is how the cell uses ATP to take things out. Once you're done with your stations, guys, make sure to complete your Edpuzzle reviews. They are extremely helpful. The cell membrane was the first one. Cell transport is really good. What cell transport will show you is active versus passive. Tonicity, that is your hypertonic, hypotonic, and isotonic. And lastly, body systems. If you have time, I highly recommend to start doing some research on the body systems. I'll put a link in the teams as well, so you can play with a little visual that they have there. But again, thank you so much for coming to Saturday school. I'll get you prepared for your exam, Unit 4 exam on Thursday. And best of luck, guys. Have a great weekend, and I will see you Monday.